Hi guys, welcome back to my channel. Today we will take a look at osmoregulation briefly. Basically, this is the nephron, and there are a lot of nephrons inside our kidney. So basically, there will be two main functions related to osmoregulation. The first one would be ultrafiltration, and the second one would be reabsorption. So let's just briefly go through what happens and describe the process in ultrafiltration and reabsorption. So this is the afferent arteriole, efferent arteriole. This is the ball of blood capillaries that make up, and it's also known as the glomerulus. This is the Bowman's capsule. This is the first coiled tubule, the loop of Henle, second coiled tubule, and the collecting duct. Of course, this picture that I drew is not accurate, but it's basically looks like this. So first of all, ultrafiltration occurs around here. So some small molecules will be squeezed out from the glomerulus into the Bowman's capsule by hydrostatic pressure, forming the glomerular filtrate. So the things that are squeezed out, the small molecules that are squeezed out, would be mainly composed of 5 things. Number 1, glucose. Number 2, amino acids, and then salt, and then urea and water. So basically, it's these 5 components, and of course, 4 of them are solutes, and 1 of them, which is water, is the solvent. Okay, and then we will have reabsorption occur. Reabsorption actually occurs in the whole nephron, the rest of the nephron, from the first coiled tubule to the collecting duct, but specific amounts and specific components or solutes are reabsorbed at different locations mainly. So first of all, we will have the complete reabsorption of all the glucose and amino acids in the first coiled tubule by diffusion and active transport. So 3 key points to note is the mechanism, diffusion and active transport, what, the glucose and amino acids, and how much completely. And while this happens in the first coiled tubule, the other 2 solutes are also reabsorbed throughout this whole thing. But beyond this point of the first coiled tubule, there will be no glucose and amino acids present anymore. So that's a very important note to keep mind of. And then after here, we'll go to the main site for the reabsorption of water. So it will mainly happen in the loop of Henle. So since the glucose and the amino acids are reabsorbed, they are solutes, as I mentioned. And you know that the concentration of the solute can be calculated by having the amount of solute divided by amount of solvent. Solute over solvent. In this case, the water will be the solvent. And since glucose and amino acids are completely reabsorbed by diffusion and active transport, the amount of solute would decrease, whereas the amount of solvent will remain unchanged. And that's why the concentration of the glomerular filtrate will decrease, and the water potential of the glomerular filtrate will increase. And that is why blood will be reabsorbed from the glomerular filtrate into the blood capillaries by osmosis. And also another key point that I always forget to mention is that you may ask, how are they reabsorbed? The nephron is actually surrounded by a lot of blood capillaries, and that's why they are in close contact with the blood capillaries and they can be reabsorbed easily. So, as I was saying, blood will be reabsorbed from the glomerular filtrate into the blood capillaries by osmosis. And then as I mentioned, the reabsorption of water will happen in the whole nephron except for this part, but mainly it will be in the loop of Henle. And after the reabsorption of the water, salt will also be reabsorbed. Again, it's the main site of reabsorption of salt. It will be also in the loop of Henle, and normally the reabsorption of salt will occur everywhere besides this point onwards. But mainly the main site will be the loop of Henle as well. And just a fun fact, the net amount of water reabsorbed from the filtrate to the blood capillaries will be 99%, and for salt it would be 80%. So, since a lot of water is reabsorbed and gone, not in the filtrate anymore, the amount of solvent will decrease, while the amount of solute will remain unchanged. And so, this will increase the concentration of the glomerular filtrate. And when the concentration increases, what will happen? Because particles will flow from higher concentration area to lower concentration area. So salts and also urea will be reabsorbed from the glomerular filtrate back into the blood capillaries. Also a key point to note, the main site for the reabsorption of urea will be in the collecting duct. But again, it will occur throughout the whole nephron beyond this point. But mainly it will be here, and there will be around 50% of the urea reabsorbed. Back to my point. So urea and salt will also be reabsorbed because the concentration increases. So they will be reabsorbed from the glomerular filtrate back into the blood capillaries by diffusion only. And yeah, that's basically it. So this is what normally occurs during inside our kidneys when it's a normal situation. So you may ask, what happens when there's abnormal amount of water or abnormal amount of salt? So, actually, this is the fun part. This is actually what happens during osmoregulation. So what I mentioned just now would just be like the normal situation, what happens involuntarily in our body. So, for example, if you have been drinking a lot of water, keep drinking, chugging water for like a long time, your water potential inside your blood will increase. And then when your water potential in blood is higher than normal, it will be detected by the osmoreceptors in the hypothalamus. And then the hypothalamus will stimulate the pituitary glands to secrete more ADH. And actually, secrete less ADH. Sorry about that. So the pituitary glands will secrete less ADH. So ADH is actually used to increase the permeability of the walls of the collecting duct to water. So that means that if you have more ADH, there will be more permeability of collecting duct to water, and more water will be reabsorbed. If you have less ADH, you will have a lower permeability of the wall of collecting duct to water, and you will have less water reabsorbed by osmosis. So, in my case that I was just saying right now, when you chug a lot of water in a short period of time, or long period, just drink a lot of water, your pituitary gland will secrete less ADH. So it will decrease the permeability of the walls of collecting duct to water, and then less water will be reabsorbed from the glomerular filtrate back into the blood by osmosis, and you will excrete more urine, a higher volume of urine, and possibly a more diluted urine. However, if you haven't drank water for a long period of time, basically you're dehydrated, your water potential in blood will decrease, and this will also be detected by the osmoreceptors in your hypothalamus, which will then stimulate your pituitary gland to increase the secretion of ADH, so that the permeability of your wall of collecting duct to water will increase, and more water will be reabsorbed from the glomerular filtrate into the blood capillaries by osmosis, and you will have less urine secreted, and possibly a more concentrated urine will be excreted. So you may also ask, why do we have reabsorption? One of the main importance of reabsorption is to prevent the loss of useful substances, for example, like glucose and amino acids. Amino acids are used to make protein and to make cells for our body. More on that later. Not later, but in another video, possibly. But then we have glucose for the basic metabolism of our cells, because they need energy by aerobic respiration, and also to maintain a stable water potential in our cells to prevent an osmotic effect, so that these cells will not burst and they won't shrink. Anyways, you may also ask, what brings about ultrafiltration? If you have already forgotten what ultrafiltration is, it will be what occurs here when small molecules are squeezed out. So actually, it is brought about by the difference in the lumen size of the afferent arteriole and the efferent arteriole. So you can see that the afferent arteriole has a larger lumen size, and for the efferent arteriole, there's a smaller lumen size. So actually, blood enters here, circle around here, and then it goes out. So this difference creates a large input but small output, and this will generate a pressure difference and create a hydrostatic pressure, which can be used to squeeze out some small molecules out from the blood capillaries into the Bowman's capsule. That's basically it. I hope you've enjoyed today's video. Bye.